Hey everyone, how's it going? Uh, Sorry I couldn't be there, um, but I wanted to go ahead and uh, talk to you about some of the information that we were going to learn this week anyway. This is really one of my favorite things to talk about. So I really wish I was there in person, but hey, this is the next best thing, okay? And so we've talked about the basic ideas behind evolution and Darwin's thinking and the step-by-step process that he used to come up with his idea of natural selection. And so, what we want to do now is just give you some examples of the evidence for evolution, the things that make us believe that this is how life progressed, and this is how we explain the history of life on Earth. So, if you take my evolution class, we'll spend 2/3 of the semester on just this topic, just giving example after example that all point toward this same idea. And you remember, in science, you've got to provide evidence. You can have a great idea, but you need some sort of physical evidence. You need some sort of observation or experiment that supports your hypothesis. So that's why all this is very important. Now, of course, we're just going to scratch the surface here. This is the 10,000-foot view. Um, I mentioned that we'll spend most of the entire semester talking about evidence in my other classes. But this just gives you an idea of the types of evidence that we use to support this idea of evolution. And one of the reasons that Darwin is remembered for this is because he was very good at collecting such evidence. And so his book is just filled with all these different examples that all help to explain this idea. And so I like to break these down. This is not my idea. I got this from somebody else. But sort of five broad types of evidence that we use to support this idea of evolution. And so that's anatomy, the fossil record, molecular biology, biogeography, and then direct observation. And so I'm going to give you a couple examples of each of these. Okay, so starting with anatomy. When we talk about natural selection, it does not create new traits. Mutations create new traits. Natural selection just takes those traits and edits them or selects for traits that are already present. And we call this tinkering. We call this taking something and modifying it, giving it a different use or making it a little bit larger, or smaller, or better, or whatever. And so the end result are these things called homologous structures. We see the evidence of this tinkering in homologous structures. And these are structures that are similar in two different species. Uh, their underlying structure is similar, but they might have very different usage. And a very good explanation for that is that they came from a common ancestor. Again, mutations are what creates these new traits, but then natural selection acts upon those traits. And so, what I was trying to say earlier is that homology is similarity resulting from common ancestry. And so, structures that are similar in two different species, and the easiest explanation is those structures were in the common ancestor of those two species, and it got passed down to each species from that common ancestor. And so the classic example is your appendages, your arms, your legs, right? So here we've got several mammals, not several mammals. We've got several mammals, but also an amphibian, um, but all vertebrates. And these are very different organisms: humans, bats, porpoises, horses, frogs. They live in very different environments. They're very different species. They use their appendages in very different ways. But when you look at the underlying structure, they have the same underlying structure: one big bone, two smaller bones, some little wristy bones, and then some phalanges or some finger like bones. And so you can see that in your arm, but if you look in the wing of a bat, you see the same thing. If you look in the flipper of a porpoise, you see the same thing: the one bone, the two bones, the wristy bones, and the phalanges in the leg of a horse or the leg of a frog. And so you can see how they're underlying structure is the same, and it's color-coded here to show you that. But of course, you can see how they've been modified, right? They're lengthened or shortened or made lighter or stouter based upon the environment that the organism lives in. And so that's an idea of homologous structures. And when we look at species, we see a lot of these. And the easiest explanation is that they inherit the bone structure, or they inherit the homologous structure from the common ancestor. And so, again, each species has a very different use for its forelimb. And so did completely new limbs, evolve to, you could easily look at an organism that lives in the water versus one that flies through the air or one that walks on land. And you could say, okay, well, there's a better way to build this forelimb based upon where we are. There's a very good engineering that could take place there, but that's not how it works. Even though they're all in very different environments, they all have the same underlying structure. The easiest explanation is that natural selection tinkered with what was already there. The early ancestor of vertebrates, which would have been like a lobe-finned fish, had this structure, and then it just got modified in its descendants. Descent with modification. Here's just another figure kind of showing you the same thing. The one bone, the two bones, the wristy bones, and then the phalanges. And you got a human, now you got a cat, and a whale instead of a porpoise, but same group, and then a bat. So again, these appendages are all used very differently, but they all have the same underlying structure, probably because they got it from their common ancestor. That's the easiest explanation. Other homologous structures we see. If you look at comparative embryology, you can see structures that are not obvious in the adult organisms, but if you look at how they develop, you go, oh, wait a minute, these are similar structures. Now, we've talked about some of these in lab. So, for example, just very broadly, if we look at the animal groups in general, right, we had the different developmental stages and the different germ layers. And so we said, all animals form a blastula. And of course, there's, wide variety in the different types of animals that we see and all the different ones that we've seen in the lab already, but they all start with that same structure. That's a homologous structure that came from the common ancestor. The different germ layers, some just have two, some have three, sponges don't have any, but that's, how the easiest explanation is that they got that from their common ancestor. Other things that happen at the embryonic level. Here's a picture of a chicken embryo and a human embryo. And if you look, they both have pharyngeal pouches or pharyngeal slits. They have gill slits. And these are the same gill slits that you see in like a shark. But they start in the embryo, but then as the embryo develops, they close up and they form different structures. They've been modified in the descendants. Also a post-anal tail. And so you had a tail at one point in your development, but you don't have it. Every once in a while, somebody's born and they still have that one. But, why? What's an easy explanation? Well, our ancestors had that post-anal tail. We still have it, but it just is greatly reduced in humans. Here's a figure from your book showing you the same thing, showing you those pharyngeal pouches. And so if you looked at adult ray-finned fish or lizards or birds or humans, they don't look very much alike. You don't see these similarities. But the more you study them and you study their entire development, you start to see these similarities. So again, these are things that came from the common ancestor. And then interesting examples of homologous structures are vestigial structures. And so we mentioned the post-anal tail. These are structures that served important functions in the organism's ancestors, but they're not as important anymore. They might still have some function or a greatly reduced function, but a lot of times they have no function. So again, why do you have a post-anal tail? That's a vestigial structure. It's not something that you use anymore. Your wisdom teeth. Most of you have had wisdom teeth and you had them cut out, right? These are teeth that erupt sometime around puberty. and they can often crowd your other teeth. They can often be painful, and so we just cut them out. We don't need them. Well, why do we grow them? Where do they come from? We can exist without them. It's because our ancestors had longer jaws. Our ancestors had more room for those teeth. And as our jaws got shorter, which gave us an advantage, for the diet we were eating, probably, we no longer needed those teeth. Yet they still show up. You're familiar with the dew claws. If you've got a dog, a lot of dogs have dew claws or other artiodactyls, even-toed ungulates. And again, you can see similar structure between like a pig or a dog, but they have these dew claws. They have these reduced claws. They don't have any function. There's no useful function for those. Or if there is a function, it's greatly reduced. Those used to be weight-bearing toes, and now they don't need to bear the weight anymore. And so they have a different use. That's a vestigial structure. It was more important in the common ancestor. Now, Darwin knew about these, and we've talked about these kind of structures for a long time. But now that we can look at the DNA, we see ridiculous numbers of vestigial structures, things that existed in our ancestors that we still have, but we no longer use. And so when that happens in the chromosomes, we can call those pseudogenes, fake genes, right? If you look at the sequence, you see it perfectly matches another gene, either somewhere else in a different chromosome or in a completely different species, except it's mutated just enough that that gene no longer works. What do I mean it no longer works? It's turned off, just like before when we talked about genetic control. That gene is no longer transcribed or it's no longer translated. And so if you find the structure, that gene is there, but it doesn't do anything. That's a pseudogene, and that's a vestigial structure. So, for example, humans, we have about 17,000 pseudogenes. Now, we have about 20 or 22 or 25,000 actual genes, functional genes. So we have almost as many pseudogenes as we do functional genes. You've got hundreds of genes for olfaction that make receptors that are used in the sense of smell. And so they make proteins, and then those proteins are deposited in the nasal cavities, and then a particular molecule whose shape matches that particular protein binds to it, causes the protein to change shape. That causes a nerve to fire. That's how you smell something. Well, we have hundreds of these genes, but about 70% of that 70% of our affection genes are pseudogenes. We have the gene, it's identical to genes that we see in other mammals, but it doesn't function in us. And so if you go and look at your dog, we can find the exact same gene, sequence for sequence, except ours are mutated in such a way that ours don't work. And so, what's an obvious explanation? And the obvious explanation is that the ancestor of mammals had all those genes and olfaction was very important sense to them. But then as we evolved, olfaction was no longer important to us. We're vision based. Vision is much more important to us. So our eyes are what we use. So we no longer use olfaction. So those genes could mutate, and it didn't really bother our ancestors because our eyes were what we were relying upon. Whereas your dog has the same genes and they still work. If you look in whales and other cetaceans, aquatic mammals, 100% of these genes no longer function because these are olfaction genes that work in the terrestrial environment. And so when those aquatic mammals moved back into the water and evolved back into the water, these genes were no longer useful. And so over time they would mutate. But again, it didn't hurt the organism because they no longer lived in that terrestrial environment. So pseudogenes are a really great example of vestigial structures at the molecular level. Now here's a great example. The teeth in chickens. Now, every semester I say the same thing. We all realize that this is a photoshopped picture, right? Do not go home and tell your parents that, oh yeah, my zoology professor said that chickens have teeth. They do not. This is a fake photo, okay? But the fact that hens have genes for teeth is not fake, right? Chickens don't have teeth, but they have the genes for teeth. Well, what's the explanation for that? The easiest explanation is their ancestors had teeth. The chickens no longer used them, so when those genes mutated, they stopped growing teeth, but it didn't harm them. And so they have pseudo genes for teeth. The interesting thing is that researchers studying these genes spliced the reason that the genes that the chickens don't grow teeth is because the teeth gene does not have a functioning promoter. And so you remember, if you don't have a promoter, then it's not going to get transcribed, it's not going to get translated, and so that gene's not going to do anything. So people studying these genes spliced in a working promoter into a chicken, raised the chicken, and the chicken grew teeth. And the teeth looked like this. They were conical teeth, like you see in other reptiles. And of course, chickens are closely related to reptiles. So again, what an interesting example of structures that exist that are no longer functioning, but they used to function. And so again, chickens do not have teeth, but they have the genes for teeth. But we see this in whales. Killer whales have teeth. Baleen whales have the same genes for teeth, but they don't function. They're mutated to where they don't grow teeth, but they have the filters that they use to filter phytoplankton, so they don't need the teeth. Okay, the next piece of evidence, or the next thing we use to help try to explain the history of life on Earth, is the fossil record. And so the fossil record is really interesting. We see, the extinction of species. We see new groups showing up. We see changes over time. We could do a whole semester on the fossil record. You could be a paleontologist, do your whole career on the fossil record. It's very well established. This is also something that helped stimulate Darwin, and this is why Darwin sort of happened when he happened. Fossil collecting was very popular in Europe, especially England, in the mid 1800s. And so a lot of people were out looking for fossils. A lot of fossils were discovered there in England, but of course around the world. People began to get this idea that these are, remnants of earlier organisms. And so how do we explain that? And so that's what kind of, again, helped stimulate Darwin's work. And so just as an example, here is the location of the great city of Bay City, Illinois. Population 6, maybe. It's where my in-laws live. And so I was up there, this is several years ago, but when my daughter and my nephews were young, we all went out to a gravel quarry that's there near Bay City, and we were just playing out there and looking at rocks and playing in this gravel quarry. Now, this is 172 meters above sea level in southern Illinois. And I found these fossils. I found that, these are called crinoid columnals. And so these are marine echinoderms. So we're going to talk about echinodermata later in labs, starfish and things. These are organisms that live in the shallow areas along the shoreline of oceans. And so why, how do I explain finding these marine organisms all the way up in Illinois? Well, that's kind of what I'm saying here. Why were they found here? And this is, well above sea level, 170 some meters above sea level. Well, let's go back to when those fossils were formed, about 50 million years ago. And the Earth looked very different. And so here's a link, and I'll put this link on the Canvas page. I can't really get it to function here on this video, but here's a still showing you. And so this is what the Earth looked like 50 million years ago. And if you look down here, I don't know if you can see my cursor or not, but just look at where I showed you on southern Illinois, and you can see that the ocean reached all the way up into Illinois. In fact, where we are in Kentucky was all completely underwater. And So that explains why I found these fossils in that place, a long way from the ocean today. But 50 million years ago, it wasn't like that. Again, I'm going to give you that website. Play with it. It's a lot of fun. You can kind of see sort of what the continents and the sea levels and everything look like at any point in time. Um, and so, again, like I said, we could talk about the fossil record for an entire semester, and there's just lots and lots of just no end to interesting stories and interesting things people have understood. This being Kentucky, we always got to talk about horses, right? And so the evolution of horses and the horse fossil record is very well developed. We found several different species, and it's very well studied. And so this figure from your book is sort of showing you the evolution of horses over time and all the different species that we have found. And you can see that, just 5-10 million years ago, there were many, many species of horse. Um, but then they all went extinct. And so just the one species that we're familiar with today is the only one that sticks around. But there were lots of others. This is well documented in the fossil record. Here's a great example, one of the neatest examples of fossils. We talk about this a lot in my other classes. Tiktaalik rosier. And so this is a fossil that sort of represents and gives us an idea of the transitional form from aquatic vertebrates to terrestrial vertebrates. And so this is when tetrapods evolved. We're tetrapods. Tetrapods are terrestrial vertebrates that have 4 appendages. And so this is a great example of that gives us an idea of how those vertebrates evolved from the water to evolve the ability to survive on land. And so we call this a transitional form. And if you look at this fossil, it has characteristics of both an aquatic vertebrate, so it has characteristics of a fish, but it also has characteristics of a terrestrial vertebrate. You see a mix of characteristics. So it's got fins, it's got gills, it's got scales. You can see the scales. You can see the fins right here in this picture. But it also has a flat head. Fish, their head is flattened laterally, but this is flattened dorsoventrally, right? And so fish's eyes are on both sides of its head for the most part, whereas this organism's eyes are on top of its head. And it's got a neck. Fish don't have a neck, but this organism has a neck so it can turn its head around. So you can see how this organism would be well suited for living in those shallow waters. And eventually its descendants became the tetrapods that, terrestrial vertebrates like you and me. Excuse me. So the story of finding this fossil is really interesting. And again, just goes to show you the types of evidence and how we collect evidence to support these ideas. Scientific theories and hypotheses allow us to make predictions. And so again, sometimes we think that, how can we make these claims about things that happened millions and hundreds of millions of years ago? But we can make predictions and we can look for evidence to support those ideas. And so when we looked at the fossil record of both fish and tetrapods, we could see that when the earliest tetrapods first evolved would have been about 390 million years ago, based upon existing fossils. And so if we were wanting to find that transitional fossil, if we were wanting to find that organism that was sort of showed how these terrestrial vertebrates evolved from aquatic vertebrates, we need to find rocks that are about that old, because that's when we think it happened, about 390 million years ago. And so researchers who were studying this looked around, looked at geological maps to try to find exposed rock that was 390 million years old. And they found some in like Pennsylvania, and so they dug and dug and dug and looked, didn't find anything. But then there also were rocks that were at the surface that were this age up in the Canadian Arctic, way above the Arctic Circle. And so they went up there looking for the fossil, and that's where they found it. And so that's just an example of a cool example of how, they predicted, hey, if we look in rocks of this particular age, we're more likely to find this transitional fossil. And that's exactly how they found it. And so the thing about the fossil record is, if you want to understand the Earth's history, you must explain the fossil record like this individual is doing. The fossil record is, um, very well established. The fossil record, of course, is incomplete. Not all organisms fossilize. Not all parts of all organisms fossilize. You're talking about billions of years of history. And so we're lucky that things do fossilize. However, having said that, there's tremendous information on fossils out there. People have found fossils all over the world, and they consistently support this idea that there were different organisms that led to today's modern day organisms. Okay. Next type of evidence we want to talk about is molecular biology. Of course, Darwin was not able to do this, but now that we can measure and study DNA in detail, we've learned a lot, and it all supports this idea of descent with modification. And so, not only can we look and see how organisms are related, because we understand how, we're understanding how DNA works, we can talk about how new traits can evolve. How do genes mutate? How can you change a gene and get a completely new trait, which natural selection can then act upon? And so this is just a whole fascinating new field of evolutionary biology. A great example is the human amylase gene. I've talked about this before. Amylase is an enzyme that helps us digest carbohydrates. It breaks down carbohydrates. And originally, in our ancestors, it was expressed in the pancreas, where that's a lot of our digestive enzymes are created in the pancreas, and then they're dumped into the small intestine, and they help to break down our food. But when studying the amylase gene, we find that we have several copies of the amylase gene. And some of those copies are still expressed in the pancreas, but others are expressed in the salivary glands. And so what started as one gene that was used to digest carbohydrate became many genes that all digest carbohydrates, but they're expressed in different places, which gave our ancestors an advantage. And so by starting to digest carbohydrates in the mouth, first off, you can digest them more completely because you start digestion as soon as it hits the mouth. But what happens when you digest carbohydrates? When you break them down into simple sugars. Simple sugars taste very good to us. And so by evolving this trait in our salivary glands, that meant that our ancestors were more likely to eat carbohydrates because they tasted good, but that's a whole different source of energy. And that allowed our ancestors to take a lot more energy and a lot more different sources of food. And so this is from a couple of the papers that studied this. And this is just kind of a diagram of where these genes are on the chromosomes. On chromosome 1, band 21 of the short arm. And so you can see several copies of the gene. 2 of these copies are pancreatic, and so they're expressed in the pancreas. That's where they're transcribed and translated. But then other copies are expressed in the salivary genes, and so they get transcribed and translated in your saliva. So our ancestor had 1 copy of this gene, and then it got duplicated. So this would have occurred during S phase of mitosis or meiosis, where we're copying the chromosomes. But of course, we talked about how you can make mistakes. And sometimes a mistake is made so that the gene gets duplicated. So you can retain the function of the original gene, but the new gene, if it mutates, it's not going to harm you because you still have that original gene. And so this happened, several times in our ancestors. So we got several copies of the same gene. Now, if you look at this gene, and you sequence it, you can compare it to other genes that we know about, and you can see at the start of the gene, there's part of a gamma-actin pseudogene at the start of each of these copies. And so when we look at the gene, of course, we can figure out where the promoter is. That's where the gene starts being transcribed. But before this promoter or around this promoter, there's part of a pseudogene that doesn't really do anything, but it's similar to a gene elsewhere in the chromosomes that are in a different species, or it's a pseudogene that doesn't function. And so all those copies also have part of that pseudogene. Well, this just goes along for the ride. It doesn't affect the expression of that amylase gene. It's just just an artifact that when the gene got duplicated, this pseudogene also got duplicated. One interesting thing is if you look at these sequences, you see where the arrows go, the direction the gene runs. One of the genes, when it got duplicated, and it got inserted backwards. So it just goes to show you how copying the DNA can make mistakes and crazy stuff can happen. You can also see that we have an amylase pseudogene. And so here's something that has the same base codes as our amylase gene, but for whatever reason, the promoter doesn't work. And so it's a pseudogene. So these are all artifacts of our evolutionary history. Now, if you look at the salivary genes, the ones that are expressed in the saliva, they have remnants of a retrovirus near the start of those salivary amylase genes. And that retrovirus is inserted within that gamma-actin pseudogene. Remember that the original amylase gene had this different gene kind of stuck in the front that was just stuck there, didn't do anything, but it got carried along each time that amylase gene got duplicated. Well, this retrovirus DNA is right smack dab in the middle of that pseudogene. And so what that did is that retrovirus got inserted into the promoter of that pseudogene. So that pseudogene had a promoter. The promoter wasn't working. That's why it was a pseudogene. It's a gene with a promoter, but the promoter doesn't work, so it's kind of sitting there. But this retrovirus got inserted in such a way that it actually activated that promoter. Now the promoter works, but it doesn't promote the actin gene because it's a pseudogene. It doesn't have the right, and it probably doesn't have enough base pairs to make a functioning gene. But it's stuck right in front of these copies of this amylase gene. And so now that mutated promoter turns on the amylase gene. But it just so happens that it mutated in a way that it turns it on not in the pancreas, but in the salivary genes. So isn't that interesting? How these things can happen in the DNA, and that's how new traits can evolve. Now our ancestors have the ability to digest carbohydrate in the salivary genes, which gives them more complete digestion, another source of energy, and it tastes good. And so now you've got a lot more energy. Now that you have this ability to eat a lot more energy and a lot wider range of food, now that brain can get bigger because this brain, needs a lot of energy and it can't grow very big without that energy, but now we've got that energy. That's fascinating. That's the stuff that really gets me excited. Now, if you look at this amylase gene, you can see that not everybody has the same number of copies. Some populations have more copies than others. And if you look at those populations, the different cultures tend to eat different foods, right? And so in this research study, they looked at cultures that ate a lot of starches, a lot of carbohydrates, versus cultures that didn't eat a lot of carbohydrates. And so, we would probably be a high carbohydrate culture. We eat a lot of starch and a lot of corn, and but they looked at cultures that ate a lot of tubers and things that but again, mostly a lot of agricultural cultures are going to have a lot more starch. And they looked at other cultures, such as those that eat mainly fish, right? Fish are mostly protein. And they compared the number of amylase copies in each of these, and they saw that on average, the cultures that ate a lot more starch had, on average, more amylase genes. That's pretty cool. But what's also cool is if you go into this more in the same study and other studies like this, they showed that other mammals that are closely associated with humans also have extra copies of this gene. So things like rats, um, dogs have more copies of the amylase gene than their nearest ancestor, like wolves, and I don't know what the ancestor of a rat is. And the idea is that those organisms were domesticated or live with humans, and we discard a lot of carbohydrates. And so there was an advantage to being able to digest carbohydrates. And so they evolved, those that got those extra genes stuck around. Another cool example of natural selection in humans involves lactose intolerance. So lactose is another carbohydrate, and it's broken down by an enzyme called lactase, and that's produced by the LCT gene, right? So very similar example to what we just talked about, but just a different carbohydrate. Now, about 70% of humans stop producing lactase in their childhood. And so that makes them lactose intolerant as adults. Um, and so if you're not producing lactase, then you can't break down lactose. And so you don't digest it, the bacteria in your gut do digest it. And they give off byproducts like gas and bloating. And we call that lactose intolerant. So this is one way. There's a couple different ways you can be lactose intolerant. Now, however, some humans have a different allele. They have the LCT star P allele, which is a functioning allele, and they do produce lactase into adulthood. They don't stop producing lactase, they keep producing lactase, and those humans are not lactose intolerant. And if you look at that LCTP allele, it's much more frequent in places that domesticated cows earlier. And so the frequency of it correlates well with the domestication of cows. And so that's pretty cool. In those cultures where they domesticated cows, they drank more milk. That's where we get a lot of our lactose. Those cultures tend to have more of this allele, and they have fewer incidences of lactose intolerance. That's pretty cool. The next type of evidence we're going to talk about is biogeography. And the person who kind of started this whole thing, Alfred Wallace, he also independently came up with the idea of natural selection. Very interesting guy. Biogeography is the distribution of species over the planet. And so once you look at different species and Wallace and other people, they try to understand why are they distributed the way they are, and then that became easier to understand once we had this idea of descent with modification. And so the continents were once all united, one giant continent called Pangea, but then continental drift has broken them apart. And we know that the continents kind of float on this sea of melted rock. And so that then helps to explain modern distribution of things like marsupials. Now, if you think about marsupials, where do we find them? We have one in Kentucky. We've got the possum. But you go down to South America, and there are lots of marsupial mammals down there. But then you also go to Australia, and they have lots of marsupials, right? The marsupial mammals, the young are born early, and then they crawl into a pouch, and then they finish development in a pouch. Whereas placental mammals like us, the young are, they gestate longer inside the mother, and then they're born. And so you've got those marsupial mammals in two very different places on the earth today. They cannot swim that far. How do we explain that distribution? Well, if you look at the history of continental drift, you can see that although South America and Australia are very far apart today, they once were connected about 65 million years ago, give or take. And that's when these mammals evolved. And we also see fossils of marsupial mammals in Antarctica. And of course, Antarctica, there's no mammals that live there today other than humans and seals and things, I'm sure. But there are certainly no marsupials that live in Antarctica today. But Antarctica used to be much further closer to the equator. And we find fossils of these organisms because Antarctica connected South America to Australia. And if we look at the distribution of many other fossils, we can see similar patterns. We can see patterns where they're distributed from, say, South America to Africa or through to India. This image kind of shows several different types of organisms. And that's how they're distributed today. Well, how do we explain that? Well, all those continents used to be connected. And that's the idea of plate tectonics, that they all are now moving apart, even though they're nowhere near each other today. Okay, so the last type of evidence that we're going to talk about is direct observation. It's what it sounds like. We have directly observed populations evolving. And there are lots of examples of this. And so I'm just going to give you, again, just going to give you a few just to touch upon these. But there are many examples where we've seen organisms evolve and things evolve. Good example is resistant bacteria. So we've been using antibiotics for a long time. The thing is we don't use antibiotics just when you're sick. We use a lot of antibiotics in our food supply, certainly here in this country. And so we give prophylactic treatment of antibiotics to lots of livestock. We give them their antibiotics in their food to try and make sure that they don't get sick as they grow. They grow in these feedlots. And so there's lots of use of antibiotics other than just when you get sick. And so it's a widespread use, and that's led to the resistance, several populations of bacteria that have become resistant to the antibiotics. And in fact, this is a big problem. Those of you who are going to get into healthcare, veterinary care, this is something you're going to have to concern yourself with, because a lot of the antibiotics that we use aren't as effective, or a lot of people are going to be infected with resistant bacteria. So we need to think of alternate ways to treat those people that have the resistant bacteria. But we also need to think about alternate ways to use antibiotics to try to reduce the amount of resistance that evolves. But this is direct evolution. So here's just an example of several different kinds of antibiotics and the percent of isolates that are resistant to those antibiotics over time, and you can see a clear upward trend. And so, again, it's not hard to imagine how this happens. If you've got an outbreak, if you've got someone who's sick, and so there's lots of these bacteria that are in their body, you give them an antibiotic. The antibiotic kills those bacteria. But what if there are some bacteria that have a mutation that makes them resistant to the antibiotic? So the antibiotic can't bind to their cell membrane, or the antibiotic doesn't get taken into the bacteria like it does for susceptible bacteria, or whatever reason. And so those bacteria are not going to get killed by the antibiotic. They're going to reproduce. Their offspring are going to also have this resistance. You are killing off all the susceptible bacteria, so you're killing off all their competition. So the only ones left are going to be the resistant ones. And then they're no longer being killed by that particular antibiotic. So you got to try a different antibiotic. And so this is, again, just a direct observation of how populations can change over time. Similar to this is that we use a lot of pesticides in agriculture, and we see lots of pesticide and herbicide resistance. And so we've got lots of organisms that have evolved resistance to these chemicals once we started using them. This is a graph showing the resistance in houseflies over time. And so each of these lines is a different kind of pesticide to kill houseflies. And so the width of the bar is the relative amount that's being used. And then the triangles indicate when we first detected resistant houseflies. And then the R is when most of the houseflies became resistant. And so you can just see this progression from DDT to HCH to chlorinate, just one after another. As the organisms become resistant, so we try a different chemical and they become resistant to that. Sometimes they become resistant very rapidly to that. And so this is direct observation of organisms evolving. The modern organisms are resistant. They descended from earlier organisms who were not resistant, but they were modified, and they are the ones that were able to survive and reproduce and pass on that characteristic to their offspring. Descent with modification. Lots of plants have evolved resistance to herbicide. And so this is one that's near and dear to my heart. That letter C there is some kind of a pigweed. And when I was an undergrad, I worked on a project looking at pesticide resistance, herbicide resistance in the Palmer pigweed. And so this graph just shows the number of unique resistance cases over time. And so once we go out and we give high doses of herbicide over and over again, eventually you're going to have a plant that has a mutation to where that herbicide is no longer effective on them. So you kill off all the ones who are susceptible, the only ones that are left are resistant, and now they cannot be killed by that herbicide. That's direct observation of evolution. Now, viruses are not considered living things, but they use the same genetic material that we do, and so they also can evolve. And this is another example of where we can track the changes of viruses over time as they evolve. And so when the COVID outbreak came, of course, COVID was very well studied. And so by looking at the sequence of COVID, we can see the changes. And if you remember that we had, different variants that became more common, and some of the vaccines worked for some variants and not others, and this is why. All viruses eventually will evolve around the vaccine. This is why you got to get a flu shot every year, is that the vaccines work for a while, but you're killing off the susceptible viruses, the ones that mutate, that are no longer killed, that your immune system no longer can attack. Then those are the ones that are going to proliferate, and that's evolution. And we've seen this directly by just studying the DNA sequence. So again, lots of examples of organisms evolving over time. I think this is the last one I've got here. Those were smaller organisms, bacteria, viruses, things like that. Plants are a little bit larger, of course. But here's another cool example of evolution in a vertebrate. And so this is a fish called the banded sculpin. So you can go to a lot of streams around Murray. You can go to LBL, and just about every stream you're going to find banded sculpin. They're a cool fish. They've got this big mouth, and they got these big pectoral fins, and they sit on the bottom. They swim along the bottom, and they're just an interesting fish. If you go, they're found everywhere. If you go to Missouri, there's a particular area where there's a cave that's got a stream running out. So the stream originates in the cave and then flows and then becomes just a regular stream. And if you look in this stream, you can find banded sculpins, just like the ones that you find anywhere else in Missouri. However, if you go up into the cave, you can find similar fish, but they are adapting to the cave environment. And so they're clearly sculpins. They clearly came from the same population. They're in the same stream. However, the ones that live in the cave, they're blind. They don't have any pigments, but their sensory organs, their lateral line and the sensory organs on their face are much bigger, all of which would be advantages in a cave. You don't need eyes in a cave, so once they mutate, it doesn't hurt you to not have eyes. You don't need pigment in a cave because there's no light. So once you have a mutation and you stop producing pigment, it doesn't hurt you. But if you do have a mutation that gives you more sensitive sensory, lateral line and other sensory organs, that's going to give you an advantage in that cave. So what we're seeing is, evolution in progress right there in that stream. That these fish that came from the same population, but some are in a different environment, and natural selection is selecting against those fish that can't survive in that new environment. So again, lots of examples where we've actually seen the evolution of organisms. So these are all just different ways that we can um, support this idea of descent with modification. We could go on and on about this, but this gives you an idea of the different kinds of evidence that we use. And so, again, in this sculpin example, you've got a new environment where different traits are favored, and we are directly observing evolution in progress. So this is only, 100 miles from Murray. Okay, well, that's it. Please let me know if you got any questions, and I will talk to you later. See ya.